Hi, thank you for having me. Today we're going to be talking about some preliminary results from our first year of the Shutterbee program. And this is a collaborative effort that has lots of involvement from students at Webster University as well as at St. Louis University. And we have collaborators at local institutions like St. Louis Zoo, the Missouri Botanical Garden, the Living Earth Collaborative, and the St. Louis um, Audubon Society, as well as the Academy of Sciences St. Louis. So all of those groups are involved in the program in some way, but my special shout out really needs to go to the students who have done a lot of the work that I'll be talking about today. In particular, Emily Burley, who just defended her senior thesis last week. And the data that I will be showing during this talk, it was from her senior thesis. And she did a lot of work this last semester summarizing all the Shutterbee data and making it into an analyzable form, and then analyzing the data and doing all of that work in R herself. I also want to give a shout out to two other students who are really heavily involved, particularly over the summer. Jessica Clonis, who will be following up on Emily's work looking at network analyses with the Shutterbee data, and Cody Dinkins, who did not work with the Shutterbee data for his senior thesis. He worked on a different project, but was heavily involved in Shutterbee over the summer. And then, of course, Nina Fogel, who is at St. Louis University in Gerardo Camilla's lab, and Gerardo Camilla himself for their contributions as well. I've been moving into urban environments, and that is in part for logistical reasons, but also in part because urban environments are incredibly important, and they're increasingly important because human beings are continuing to grow in population size, and we're increasingly focused in on urban environments. So more than half of the world's population are now in cities or in urban environments, and we're expecting that to continue to grow. So urban environments are really important for the future of conservation. And we often think of urban environments as being kind of devoid of biodiversity. And I love this picture. This is a picture from James, and I use it all the time, but it is one of my favorites because this is one of our favorite bees. This is Bombus fraternus, and it is only found in a few locations in the region. Although, James, was it just this last year or last year and a half, you've tracked down a few more populations, right?
Yeah, I spent a good amount of the summer in my free time tracking this guy down, or the mainly the females of this down around the St. Louis area.
Yeah, so there are actually more populations than we once thought, but this species in particular is not super common, but it does still occur in the St. Louis region, and it seems to be picked up mostly where we have relatively high quality or quasi high quality prairie or some prairie-like habitat. And so with these, so I use that as an example, but it's also an example of a broader trend, particularly in our region. So St. Louis is a cool location to be because we're ecologically speaking, right? Because we're on the line of multiple different ecozones that kind of come together. So we have pretty high diversity of plants and bees, and other organisms too, relative to our area. And so in the city of St. Louis, and this is work out of Gerardo Camilla's lab, they've documented 207 was the most recent count bee species in the St. Louis city area. And that, now not all of those species are common, of course, but that's still a really good proportion of the 452 species of bees in Missouri. So we have, within this very small area relative to the whole state, we have a lot of bee diversity. So we want to be able to maintain that going into the future and understand why, okay, we're able to maintain them to some degree, but that just because they're here does not mean necessarily that they are common. Some of these are declining, and we see filters in urban environments. We see that certain bee species are more likely to be represented in urban environments. And so urban environments, while they can support biodiversity, they act as filters. So probably not too surprising once you think it through. Urban environments have a lot of concrete. right? We get a lot of concrete, a lot of asphalt, a lot of hard surfaces. So we don't get very many bees that are ground nesters, right? Whereas we have a lot of bees that are nesting in cavity nesters or stem nesters, we see more. And in those of you who are in the Shutterbee program may have seen some of these slides and heard me talk about these things before. But I also just wanted to set the framework. And one of the other things that we find is that species with narrower niches or narrower foraging breadths, they are declining and they are less likely to be supported in urban environments. So this figure right here from 2019, a paper by Wood and colleagues, looks at the standardized dietary breadth score. So basically what that means is how generalized they are. Do they go to lots of different kinds of species, or do they go to just a narrower subset? Now, bumblebees in general are more generalized than, say, a hibiscus bee or something along those lines. That's very highly specialized, but they still within the group vary in how specialized they are. And this is the change in geographic range here on the y-axis. And you'll see that those species that are declining are also the species that have a pretty narrow breadth of the plants that they will forage from, versus those that are more generalized. They're doing okay, like Bombus impatiens, Bombus bimaculatus, Grisiacolis, those are ones that we see not ubiquitously, not absolutely everywhere, but they're very common and they do quite well in urban environments, versus these other species down here, including two that we have in this region, Bombus pennsylvanicus and Aricomis, are more specialized, and we actually see them in lower abundance here as well. One species just to point out, of course, is Bombus affinis. We do have a historical record of Bombus affinis in the St. Louis region, but the last known collection was 2008, I believe, and that was oh Lord, I'm blanking on his name. He worked at the Missouri Botanical Garden and did a bunch of collections, and he worked at Litzinger.
Richard Kleinbell.
Thank you, Richard Kleinbell. Thank you for that, James. So Richard Kleinbell had an identified one, and he did not pin it. He knew what it was, so he let it go. But he was a fantastic entomologist, so we know that he identified it correctly. So it hasn't been seen in over a decade in this region. So our yards, parks, and farms could actually be part of the solution. So I mentioned yards, parks, and farms mostly as any area that we manage as humans, any area that is either privately owned or owned and managed in a meaningful way by humans. And we often think of our small itty bitty plot of land, if we even have one, right? Maybe we're living in an apartment and we have a plot at a local community garden or something like that. We often think of those as too small scale to matter, but they do matter, and they can matter quite a bit, particularly when you add them all up. There was a really interesting paper that came out. It's actually pretty old now. I don't know if there's been, I searched for some updated estimates of this, but wasn't able to find any. But lawn cover within the United States is over 400,000 square miles. So 128,000 square kilometers. That is a huge amount of area that is just taken up by lawn. And most lawn is essentially, from my perspective, one step up from concrete in terms of how well it manages water, as well as the resources that it provides for bees and wildlife. But we know that species richness can actually increase with urbanization, particularly if we are in areas that are a little bit more semi-natural. So if we could take our garden plots, right, these areas that we think of as our own little managed areas, and if we attempt to make them a little bit more natural, we might actually be able to boost the amount of species richness, in this case bee species richness, that we're able to support. And that really emphasizes this idea of bringing conservation home being important to the future of biodiversity, because the vast majority of land is independently owned in the United States. And a lot of that is not necessarily owned by small plots of land, but as I just demonstrated, those small plots of land also add up to quite a bit of area. And there's some really cool backyard conservation programs like the Bring Conservation Home program through the St. Louis Audubon Society that we actually collaborate with for this project, where it encourages people and gives them tools to increase the sort of conservation status or the ability of that plot of land to support biodiversity. So that leads us to the main research goal for this project, this Shutterbee project. There are other goals like outreach goals, conservation goals, and I'll talk a little bit about those later on. But I wanted to focus in on the research goal for now. And this is to understand if and how landscape management affects bee diversity and foraging behavior. And said another way, what we're trying to see is we know that in urbanization influences diversity, right? Can management at small spatial scales help to weaken those filters? Does that make sense? Everybody give me like a heads up or a nod or a something if you feel like you're following me on this. Okay, great. So what we've done in order to do this and to reach our other goals of the project is to invite anybody who is interested. We pulled specifically from, or we solicited folks who are in the Bring Conservation Home program, but then we also branched out and asked people from a much broader community, which is basically anybody who's interested. And we weren't planning on doing this during the year of COVID. It just happened to coincide. And so we had a really good large group of really devoted and interested people. We had 168 people who were trained by us in the program and who contributed photographs to the program, which is way higher than we were anticipating. And we had to switch all of our training remotely. And I think it actually ended up working out really well. While it would have been nice to be in person in some ways, we tried to make it as interactive as we could. We had great groups who were really able to maintain, focus and interest in the project throughout this online training. And it worked actually really quite well and gave us encouragement that maybe this is something that we could take even beyond St. Louis. Now, for now, we're staying in the St. Louis area, making sure that we get these data and do the project well here. But it's encouraging to see that maybe some of these things could branch out and we can test some of these questions more broadly. Okay, so just to give you a little bit of forward thinking about where these data are going. So to give you an idea of the roadmap, if you will, I want to describe our two main predictions that we'll talk about today that we were testing with the Shutterbee data. So the first one is if overall flower densities decrease with urbanization, generalization will increase. And this is because as bees have to travel further in order to get to resources, we would expect them to branch out and basically visit whatever flowers they can find because it's harder to find them. Now, there's an alternative hypothesis that I'll tell you about later, because of course, as things often go in science, our predictions were not exactly supported, although they were partially. So anyway, teaser there. Prediction 2 is that conservation status will weaken the effects of urbanization so that we would actually not see the strong patterns that we would predict from prediction 1. All right, so how did we do this? Well, we invited folks to do backyard bee photographs or bee surveys and to upload them to iNaturalist. And we asked them to give us 3 key pieces of information. 1, that they were using our protocol, that's SBEE here. Their effort, their collection effort, so how long they actually conducted their surveys. And we ranked this based off of the size of their yard. So if they had a smaller yard, they were supposed to observe for about 20 minutes. And if they had a larger yard, they would observe for 30 minutes. And then if they knew it, we asked them to tell us their plants so that we could actually look at who was visiting whom and how their visits might have changed with urbanization. Our first training workshop, though, before they actually did any work in iNaturalist, was just to start building community and building confidence in knowing what a bee really looks like. Because people had very different degrees of knowledge about bees. Some people knew that they were really, really diverse and come in all kinds of cool colors and shapes and sizes. But most people, when they think of a bee, they think of a honeybee or maybe a bumblebee, right? And so we really worked with them to try to differentiate our native bees, like you're seeing these two bees here, from honeybees and bumblebees, and to start to recognize that we have features of bees that help us tell them apart from other insects, but that just because you see something that's small and black doesn't mean it's a fly. In fact, it's very likely a very small bee. And so we go through this process of how to ID a bee from other things and start building their confidence about what is and what isn't a bee. And then in the second workshop, that's when we get down to the nitty gritty and we really talk about explicitly what we want their photo surveys to look like and why we want it to look like that, right? So we need to have a standardized approach so that when we're looking at people's data, we're comparing apples to apples, right? And not apples to pomegranates or apples to broccoli or whatever, right? We need to make sure that we have the data that is comparable across sites. So we talked to them about how to construct their walking surveys. This is one sort of hypothetical path that somebody could walk through their yard. We talk a lot about how we return to this idea of diversity in bees and how they vary a lot in size. And it's not just the big guys that we're interested in, but we really want the little ones too, because we're interested in looking at bee diversity more broadly than just at the bigger bees. And then, of course, we have to control for the things that influence bee behavior, things like how sunny it is, what time of day it is, how hot it is. And so all of that helps us to standardize these surveys, despite the fact that we have people with very different backgrounds in science actually contributing to this program. So here is a map illustrating some of the locations that we were working in. So all of those red dots are the general community, meaning they're not part of the Bring Conservation Home program, but they are enrolled in Shutterbee. And then we have those blue squares, which are locations where they are involved in both Shutterbee and BCH. And then Nina Fogel, was also conducting netting surveys. I'll talk just briefly about that. I won't be able to show you data yet today, but I'll tell you a little bit about why that is important in a second. And then we have locations that were BCH that were not netted by Nina, but that were involved in the Shutterbee program. All right, so what we can do with these kinds of data is see whether or not our Bring Conservation Home locations, particularly those that are ranked as gold or platinum, meaning that they have reached some conservation status according to Bring Conservation Home, whether or not their bee diversity or bee behavior is different than what we see elsewhere. So for each of these houses, we have a little bit of localized, or each of them has their own localized resources available for the plants, and then a different level of urbanization. And we sort of use that level of urbanization to get a sense for the area around each location. So this was done in collaboration with Trey Hall, who is another graduate student in Gerardo Camillo's lab at SLU. And he has this amazing algorithm that he has written in order to get estimates, really fine scale estimates on the amount of impervious surface, the amount of tree cover, how much quote unquote grassland there is in an area, today we'll be focusing on impervious surface estimates. All right, and then as I mentioned, Nina also went to a subset of these locations with her field assistant from the summer, and they conducted surveys, a total of 65 houses they surveyed at, and they went about one time per month. They collected 3,500 bees, and they're still processing some of these to get them down to species and to genus, but what this gives us is a really good reference, a really good way to say, are our Shutterbee data giving us the same information as the data that we're getting from traditional netting surveys, right? And as I mentioned, I won't be showing those data today, but maybe I can come back in a few months or in a year and tell you more about how those data have come out. But we haven't analyzed that yet because she's still processing some of her samples. All right, so to date, we have 8,209 observations that have been coded for Shutterbee, and we have been able to identify almost 90% of them to genus or lower, which is really exciting. And I had a fun meeting yesterday with John Asher, who is a globally renowned entomologist, and his emphasis is in bees. And we were talking about iNaturalist, and he really thinks we can get down to subgenus for a lot of these groups as well. And so he was very excited about our project and the ability of us to get photographs of even these small bees that get us down enough to genus. Now, we're not going to get to species for everything, but we can get to genus, and that can give us some meaningful information. So we get them to research grade. All of these observations are verified by at least 2 members of our research team, as well as people who are on who are in the iNaturalist community, including John Asher, which is where that conversation's initiated from. But in addition, there are folks from the Missouri Department of Conservation. There's also folks who are just interested iNaturalist users and bee enthusiasts. James Faupel is another person who has been really helpful with helping us to identify these bees, because that's a lot of bees. That's a lot of bees to get through, particularly when a lot of these identifications are started being done by undergraduates who have very little entomological training, and they do a great job. So these are the bees that we ended up using for our analyses that I'm going to show you today. Apis mellifera, the European honeybee, Bombus impatiens, or the common bumblebee, the 2-spotted bumblebee down here, Bombus bimaculatus. I'm sort of going in reverse counterclockwise, Xylocopa virginica, the carpenter bee, and the brown-belted bee, Bombus griseocolus. Now, you may, if you remember from earlier this semester or earlier this talk, you may remember that these were actually pretty generalized bee species. So, sadly, interestingly enough, our specialized bees were underrepresented. And that already is pretty striking. So we ended up having to filter out about 2,500 bees for various reasons that I won't go into now, but I can certainly answer questions about it if people are interested. But out of 5,000 bees, we did not have enough individuals recorded for these bees in order to get an estimate for how generalized or how specialized they are. So Bombus auriculmus, which is this one right here, Bombus pennsylvanicus, the hibiscus bee, and the squash bee, most of them we only had enough photographs at maybe one location in order to get estimates for their generalization scores. So that right there is a little telling, I think, in that in these urban environments, we have really high densities or relatively high densities of common or generalized species, but low densities of those species that are more specialized. So unfortunately, our analyses are sort of biased towards our generalized species already, but we are hoping to continue this project for the next few years, and we're hoping to be able to pick up on more of these less abundant species so that we can we can see what the patterns look like for them. So in general, the bee behavior didn't change very much. So for bombus impatiens, for instance here, this is percent impervious surface, that's on the x-axis. Simpson's diversity is on the y-axis. That's just one metric, one way of estimating foraging diversity in bumblebees. And for those of you who are familiar with thinking about diversity, maybe this won't matter so much, but I just want to clarify a little bit to be careful that diversity is different than richness. So richness is just how many plant species they visit. Diversity looks at how many species they're visiting, but how frequently are they visiting them? Are they really visiting one species more often, or are they visiting lots of different species equally? And so regardless of rank, whether or not they had a high rank, which is in this blue color, a low conservation rank, which is in the purple, or whether or not they were unranked, either in the BCH program or not, we don't see any patterns. There's no change in their behavior. They are relatively generalized, and they're just visiting any old plants they can find. We see the same thing for Griciacolis as well as Xylocopa virginica. So all of these relatively specialized bees that I'm showing you here aren't showing a pattern at a sort of intermediate spatial scale, which is at 1,000-meter buffer. So that means that our impervious surface here was estimated for a 1,000 diameter circle around the location. However, when we scale up just a little bit more, we get an interesting result for Bombus griciacolis. Now, unfortunately, conservation rank doesn't really seem to matter. So there's no effect of conservation rank, but for Bombus griciacolis, we are seeing an effect of impervious surface. So the more impervious surface there is, the more specialized they are. Now, if you recall, this is actually the opposite of what we predicted. We predicted that as impervious surface increased and therefore flower densities decrease, that we would see these bees really needing to branch out, visit more flowers in order to get the things that they need. However, we're seeing the opposite trend here. And this is, if you'll recall, I mentioned an alternative hypothesis. And there's this really interesting alternative hypothesis from the foraging literature that's a bit more on the behavior side of things versus the community level side of things. And what they showed in some of their work is that species, when they have to work harder to find things, they have to search harder in an environment, then they may specialize on the plants that they actually will visit, because they're expending so much energy just trying to find the plants that then once they get there, they're focused on the ones that they know how to forage on. And essentially, I think that might be what we're seeing here. As impervious surface increases, as flower diversity or flower densities start to spread out, right? We get these patches that are spread apart. These bees are sort of narrowing in on a suite of species because it takes them so much energy to search through the environment to find them. There is, of course, an alternative hypothesis that potentially diversity of the flowering plants is just lower. So that's something that we want to explore in the future, whether or not it's a direct effect of their response to the plants that are available, or if it has to do with the regional patterning. Now, this data would suggest that it is the regional patterning because we're not seeing a difference even when we have locations that have high diversity of plants. So we're not seeing a difference by conservation rank. Apis mellifera is doing something different, and it's doing more what we would expect, what Emily had devised as her prediction for her project. They are more generalized in urban landscapes. And interestingly enough, they're also more generalized in high conservation status locations. So what that means to me, when we combine those two pieces of information together, this implies to me that when their plants are available, they will forage from whatever they possibly can. And as impervious surface increases, they have to go farther to search for these things. They're more likely to generalize. So we sort of have essentially two different mechanisms happening at different spatial scales. And once we have that high diversity in plants that are available, despite the level of impervious surface, these guys are really generalized. So our BCH unranked and our SBEE unranked, those populations, you'll notice, or the bees that are in those locations foraging were more specialized than those that are in the BCH ranked houses. So that's really interesting because Apis mellifera is not native. It is a very highly generalized bee that is of European and Eurasian descent. And so it almost appears as if the conservation practices are doing more for honeybees than they are doing for anybody else. Now, these are very preliminary analyses. We have a lot more analyses to run, but that was something that was a little bit surprising to me. I wasn't expecting to see such a great impact on Apis mellifera since they're already so generalized. I was expecting to see much greater effects on the other species. And again, this may relate to flower diversity in those locations. These are counts from Nina Fogel's work, not last summer, but the summer before, looking at BCH homes, depending on their ranking, either high, low, or unranked. And not too surprisingly, these locations that are ranked high with diverse or high in conservation status have higher species richness. And so this is just supporting that idea that the honeybees are responding to local floral resources availability, whereas Bombus grissiocolis isn't. It really seems to be responding to these large scale patterns in resource availability. And they're really the only two species that we've documented a response for at this stage. So that's really interesting. And I thought here would actually be a good place to pause. I have a little bit more to say about other aspects of the program, but I thought we'd maybe pause here and I can field some questions about our results to date. Any questions? Feel free to unmute yourself or put them in the chat.
Nicole, I have a question. This is Jim Jordan. I participated in Shutterbee, and I found that certain bees were very, very difficult to photograph. And so did you get a sense that there were missing genus or species as a result of them flying off even before you had a chance to capture them?
Yeah, we haven't done those analyses yet. I am anticipating that that is going to be not an uncommon experience. And so that's one of the things that comparing with Nina's data, we'll be able to say which groups this seems to work well for, and maybe which groups it isn't currently working as well for. And my hope is that we can try to improve our ability to pick up on those species through time, with better training on our part, as well as practice on participants' parts. And, you know, one of the things that we did was we had people going out and taking photographs every 2 weeks, right? Whereas Nina was only going out once a month. And in part, that is to make sure that we're not missing anything phenologically, so going through time, but also because we recognize that the effort in order to pick up some of these things for photographs might need to be a little bit higher at each location because of that bias. So we'll see. The jury is still out on that. But that is something that we have heard repeatedly. You're not the first person to have had that experience.
Yeah.
Now, most of the species I've shown here today, that wasn't an issue for. These are all the big bees. So, you know, these, if anything, were overrepresented. And in fact, this coming year, we might say, all right, after you've taken a few pictures of your bumblebees, move on unless you see a new kind of bumblebee. Because we don't want to short shift the small guys because we see so many big guys. And we encouraged that a little bit in the training, but I think we could probably do a little bit better job of that in the future. Other questions or thoughts or, yeah, surprises? I'm happy to hear feedback on interpretation too, you know, anything. Not right now? All right. Well, we can move forward then, and I can talk about something else. If other questions pop up, please do share them. So just to summarize those results, most generalist species don't exhibit much of a change with urbanization. Now we have very generalized species, so we'll see, hopefully in the future, we'll be able to see what it looks like for more specialized species, see if we see any differences for them. And those species that do exhibit a change exhibit contrary patterns suggesting that they're responding to different spatial scales. And that's pretty cool and interesting and exciting. And from a conservation perspective too, right? Because it tells us a little bit about where our efforts maybe need to be focused on, for certain groups anyway. So our next steps are to, as I mentioned before, accumulate more data to test the effects on those rarer, more specialized species, and improving training so that we make sure that we get those rarer, more specialized species. And then what I've shown you here is really a very narrow subset of the observations that we have for these communities. We wanted to look explicitly at behavior, primarily in the large bees, and that was Emily's interest for her thesis. But our next step is to really look at the whole community and look at who's interacting with whom in a much broader scale. So these kinds of what are called network analyses, although they're misspelled on that slide there. I have a transposition of two of the letters. But these networks give us information about who's interacting with whom. And you can take into account that you have species that were not collected very frequently. So unlike when we were looking at generalization scores, where we really had to exclude a lot of species in order to get the replication that we need, those species can still be included in the networks. And so what this will tell us is the structure of the communities different? And that can give us some key information about the pollination services that those bees are providing to the plants. And on the flip side of that, how the plants are providing resources to the bees. And then there's another suite of theory where you can actually manipulate the networks and sort of say, okay, what happens when we remove this species? How much does that affect the whole structure of the community? And that can tell you a little bit about the resilience of the community. Now, it's just a modeling procedure, so it is not necessarily predictive, but it gives us some information about how flexible the system has to be in order to respond to greater or increased disturbance. All right, so on to just a few other goals of the program. So one goal, it's an outreach program, right? I mean, this is a community collaboration. And so we really want to engage with the public in conservation science. And we try to do this in a way that's meaningful. Many of you are in the program. So please, if there are other ways that we can be more meaningful or make your experience more enjoyable, please do let us know. And, you know, one of the things that we want to do is open lines of communication between those of us who are working in conservation science and those of you on the ground actively manipulating the landscapes in certain ways, either for conservation purposes or gardening in your backyard, and really try to educate both sides. So, demonstrate the great diversity that's in the area, talk a little bit about conservation strategies for helping to support bee diversity, but then also from our end, learning about what's in your yards, learning about things that you know about what has worked and what hasn't worked for your yard and how your yard fits within this whole network. So there is a question that just jumped up in the chat here before I move on. This is from Ellen Hartz. I am a BCH participant and a backyard beekeeper. I know I am ineligible for participating in your program, but would like to be able to be trained as an observer. Would that be possible the next time a training is offered? Yes, that is definitely possible. And one of the reasons why we were removing people who have managed hives is that we know that honeybees interact with bumblebees and other small bees on the regular. And so if we have managed colonies, that can influence the patterns that we're seeing. And so since we were really interested in trying to understand not necessarily how honeybee hives influence behavior, we had to exclude those locations. However, yes, you could absolutely be trained as an observer. We could choose another site location. Once LREC is open, maybe that would be one of your locations, or we could choose another location too if you work by a park and you're interested in doing it on your lunch break or something like that, then we can find another location. Absolutely, that is something that would be great. And might actually be able to fill in some gaps in other ways in our data. So yeah, absolutely. So we try to communicate with our team, our whole research team, meaning all of our participants and us. And in order to do that, we have a really great team of, or this last year, we had a really great team of undergrads, Emily Burley, Cody Dinkins, and Jessica Clonis, and Nina Fogel, who I've mentioned a few times now, and Gerardo Camillo, and I all interact with folks in a myriad of ways, either via email or on social media or through our newsletters, and just trying to continually share the information that we are learning and providing additional conservation ideas and give quick feedback on training as well, as a way to continue to maintain communication, because no collaborator likes to be left in the dark for like a year about what's happening with their data and not knowing what it's meaning, right? So we're trying to continue that conversation, and we share latest science news on bees and things like that. That includes a bulletin that we send out, as well as some training materials, and a bingo game that was really fun. I think we have to adjust our cards next time a little bit, but that was our first year doing it, and that was really fun. Not everybody took to it, but some people really did and had a good time with it. So basically looking for the bees in their yard to see if they could get bingo. At the end of the year, we did a survey. Now, this isn't a research survey. This is like a program assessment survey, just to see what what was driving our participants to be involved, what they wanted to learn about, and so we're going to take this information and adjust our program a bit. We had more than 50% of people who really wanted to learn more about pollinators and were really interested in learning about the organisms that are living in their environments. About a quarter of them were interested in contributing to science. And these are not necessarily mutually exclusive. Some people were interested in learning about one thing or the other. This is just like their major, their top. 4% were just excited about learning or using iNaturalist. 5% were personal enjoyment, getting out in their yards and being part of their environment, and then 13% were really interested in learning about plants. So we're going to build this up. We're gonna try to beef up our learning or information that we're sharing about bees and about pollinators. And one thing, if you're interested, this is another way that you could get involved too, Ellen, potentially in the future, or somebody else who doesn't necessarily have a good location to do observations. We are, when it comes to identifying these species, we had 168 people taking the photographs, and we had about 10 people regularly identifying. We had more than that as identifiers, but 10 really regular on there helping us move these forward. And so one of the things that we're going to do this next year is to invite participants to become sort of certified identifiers. And not that you will necessarily know every single bee, but we're going to cover some of the really common bees first, so that you get to know the big bumblebees. You can help us process some of those photographs, get to know those species really well, and then eventually maybe go deeper with other species that are a little bit harder to learn. So if that is of interest, that's something that we're going to be branching out here in the next year. We haven't launched the program yet. But we are going to have a Shutterbee bee Photoshop or photo identification workshop, although maybe we should call it the Shutterbee Photoshop, although there's probably proprietary reasons why we shouldn't do that. Anyway, we're going to be doing that this January. So if you're part of the program, look out for that. We'll be announcing that in our bulletin coming out before the holidays. So here are some of the education materials that we have shared. I sort of already talked about this, but in addition to what we're doing for our participants, we are also creating educational materials for school-aged kids, and this is another way that James has been involved in the program, as well as educating adults through our ID photo workshops and things like that. So that'll be fun. I'm really excited about the next step with that. And last but not least, if I still have time and I'm not boring anybody too much, we are also collaborating with Amy Nedlasky and Aaron Tate from the St. Louis Zoo, and to look at whether or not participation in community science changes conservation behaviors. Now, we recognize that this program, for people to be involved in this program, they're already going to be to some degree committed to conservation behaviors. And some people are going to be very committed already, right? People who are BCH homeowners who have a platinum ranked yard, they've been very thoughtful for many years about modifying their landscape to support biodiversity. So we're not necessarily expecting to see changes in behaviors in those people, but they're like the people that we're trying to emulate, right? That we're trying to get people to be aware of their environment and to see whether or not behaviors in those people who have less experience in conservation biology might be influenced by their participation, or might not be moved by behaviors or by their participation in these programs. So let me finish this up just a little bit here, and then I'll move on to the chat. I see there's questions coming in there too. So we're not the first people to think that maybe involvement in community science could influence people's behaviors. There's been studies that show that it can, but they tend to be not hit or miss exactly, but not grounded in theory. And one of the things that we're trying to do with this research, and this is research that's definitely spearheaded by Amy and Aaron at the zoo. They are the experts in this, not I. But we've grounded it in the theory of planned behavior. And this theory posits that or suggests that our behaviors are really driven by three key things: our behavioral beliefs, our normative beliefs, and our control beliefs. And each of those things influences our intention to act and then our actual behaviors. So one of the things that we're trying to do is to understand at a very sort of mechanistic way, if you will, how participation in community science might influence behaviors, conservation behaviors in general, and how that might, it wouldn't be too surprising if somebody came from this program and they were like, oh, okay, well, I'm going to increase plant diversity in my yard so I can support more bees. And that's important and that's interesting, but that's not surprising. The question is does that level of change in behavior or belief or agency, does that cross domains? Does that also make people recognize the importance of their yards for water management and for other things that are related, because this ecosystem is an interrelated, complex system? So that is, we actually did a retrospective with this, meaning we had people in Shutterbee. Those of you who are in Shutterbee received these emails asking you to participate in this program. It's technically a separate study under the umbrella of Shutterbee. So you were asked, in other words, if you're not interested in being involved in the behavioral theory, but you still want to do Shutterbee, you totally can. But we invite people who are in Shutterbee to be involved in this other program as well. And so we have multiple groups that we are surveying attitudes and behaviors over time. This looks very, very complicated, but what it really comes down to is, are they in BCH? Are they in Shutterbee? Is Nina working there or not? So we have some people who are in BCH who are not part of Shutterbee and not part of, there's no bee things going on from the research perspective on their land. And that's going to be a big comparison between this group and this group, as well as this group right here who are not involved in BCH but are in Shutterbee. So to see whether or not we're seeing any changes in behaviors over time in groups depending on their involvement in community science. So that'll be really interesting future work. So going to this question of, do we have a website and how can we get involved? There is a mailing list. So if you are interested, or if you know somebody who is interested in being involved at any one of these levels, please do shoot us an email or go to the website. The Shutterbee website is currently housed as a unit of my website. So you can also search for me and you will find that website. We have a tiny URL for it just so that it can be found a little bit more easily. But I see Dean has actually put in the link as well in the chat if you want to go there. If you want to be on the listserv that sends out the bulletins and things like that, you'll get notifications and news about the program before enrolling in next training, which won't happen until the spring or late spring probably. Then please shoot me an email and I would gladly add you to that list. The Shutterbee email goes to both me and Nina. I'm usually the one who mans it most of the time though. So that's shutterbee@webster.edu, easy to remember. And you can contact me at any time. And with that, I think I'll stop for now. And yeah, I'll open the floor up again for any questions, thoughts, concerns, give me your input. Yeah, so Scott asked if we have thought about having our Shutterbee participants net once a month. Yeah, potentially. What we want to do is see whether or not that's a feasible idea. Are we going to get good data that way? Are we going to get enough data? Because the photographs tend to be at a lower density than the netting. So we could potentially increase the observation time if that was the issue. But for now, we're still planning on doing it every 2 weeks and then telling, if people miss a week because they're on the vacation, well, that happens. That's life. That's not a big deal. But for now, we're not going down yet because we haven't looked at the data, and we'll really need to model that and say, okay, if we were to only select this number of observations, do we get similar data to what we see? That was in addition. Net once a month. Oh, that's a great idea. I'm sorry, I misread your question completely. I missed the net. So yeah, I mean, if we have people who are trained as, and used to netting, that would be fantastic. That would be a great idea to have a subset of people. We've been netting ourselves, and we're planning on doing that again this year at a subset of locations. But if people are interested in that, yeah, that's something that we could open up. That's a great idea. Scott, are you offering? How critical are photography skills? Oh yes, great. That'd be amazing. Yeah, that's great. I'll keep that in mind. I'll write that down. I'll write your name down, and I'll think about that.
Well, with that in mind, Nikki, would you then be having them like put them in, a cooler to just cool them, photograph them, and then release them alive, or would they be collecting them and have to train them how to pin and all that?
No, we would do the photographs and release. I think because that's what we're planning on doing anyway at a few locations. So Nina will still do her netting, where she's going to go down to species level and really get the high. And if somebody is already trained as an entomologist and wants to do that, then they can. But I think probably the best thing would be to train them to do it on ice, get photographs, and then release. I really try to reduce lethal sampling as much as possible. So I know that isn't always feasible for the smaller bees. I'm trained in bumblebees, so I really, you can take photographs of bees and release them and get what you need. So I don't really want to encourage lethal collection at all of these locations anyway. So how critical are photography skills and equipment to success in this study? Well, certainly having photography skills and equipment helps, but we have a lot of people who don't have much training. And it's really more about how comfortable you are using whatever tool you decide to use. A lot of people use their phones. You can get some really decent photographs just using iPhones, Androids, what you're usually used to, many of us anyway, are used to using, right? This is what we go if we're taking a walk or something like that. We see something cool, we take a picture with our phones because they're with us a lot of times, at least not everybody, but a lot of us. Now, that being said, there is a little bit of a learning curve about taking pictures of moving organisms, right? And during the training, we try to help out with that and provide some tips. We are not photographers, so when we do have photographers that are around, we ask them to sort of give their tips on taking pictures of moving things. But sorry, this is what happens. I get distracted by the chat. So anyway, no prior experience is necessary. I will say one thing that has hindered some folks is just not having quite the eyesight to be able to see outside on their phones. That is maybe the major killer at that stage. Then it's really hard because it's very frustrating for them because they feel like they can't take good photos of bees because they can't see well enough outside. So that would probably be the only limitation. But if that's not an issue, then you're good to go. Are we going to send a post-season evaluation of to the Shutterbee participants? Tina, I'm guessing you mean about their surveys, like how they did with their surveys. Is that what you mean? Just to clarify. If that is what you mean, we had not planned on doing that, but that is a great idea. We were planning on in the spring having a review with everybody, inviting all of our participants who want to participate again and just say, here are some common things that we saw. Here are some ways that you can improve to do those. So one thing that we know some people had issues with at first was uploading duplicate pictures. I don't know if they just weren't sure if it went through or what, but we would end up with duplicate images, and that throws off our counts, so we can't have duplicates, so things like that. We weren't planning on doing it on an individualized basis, but if there is enough interest in that, then certainly we can. We've had a couple people ask. So maybe the best way to do that is just to send us an email and let us know if you want your individualized sort of, how did I do? Sure, Cindy, I can definitely give some quick tips on photographing bees. Yeah, they can be skittish. Sometimes they're not. And it depends on the season, it depends on the species, it depends on the cast, right? Queens early in the season of bumblebees tend to be a little bit more skittish than the workers later in the season. Some of the small bees tend to be more or less skittish. So a few of my top hints or tips. One, use a live or burst function, because there's always a delay between when we press the button and what we're seeing, right? So there's that mental processing of, hey, body, touch the button. I want to get this bee picture, right? So that is one that's really helpful. The live function in particular is kind of cool, if a little bit creepy. So basically, when you have your photo app open, it's always taking a small chunk of video, and then when you take the picture, the live feature saves a little bit before when you press the button and after when you press the button. So that's pretty handy. And then you choose the frame that has the best angle. Another one is try to have the sun coming at an angle to you, not behind you, or really not in front of you either. The best one is having it kind of at a side angle so that you can get good reflected light. And so if the sun's behind you, you're blocking the light from the bee. If the sun is behind the bee, then you're kind of blinding your camera. So that's another one. Let's see. For not scaring them, what we ask people to do, we ask them to do walking surveys, because we don't want you to stand in one location and miss what's happening everywhere else in your yard. However, we do say to walk really slowly and to pause as you come to a group of flowers, sort of sit for a minute or 30 seconds. And the bees usually end up getting a little bit comfortable and will continue to forage. And so you can snap some photos around them. Another thing that I do is I'll try to take a photo that's a little bit farther away so that I know I don't miss the bee, and then I'll move in closer just in case they fly off, and take pictures as I get closer. Let's see, other ones. There are going to be some that are just wicked fast that you're not going to get very good pictures of. So if you miss some, forgive yourself. That is just like the same way that we miss bees when we're netting. It's just the nature of the game. Let's see. James, do you have other suggestions? You can get these really cool, like, magnifying glasses, but you want to make sure not to get the super high magnifying glasses that clip onto your phone, but you can get ones that maybe go up just 5 times, and that might be helpful. Anything that's 10 or 20, way too high to get good photos of bees flying.
Yeah, the one I use is a 10. And so I was going to say this actually works pretty well because the camera is like your little bee in front of me. And I was going to show, taking a picture of a bumblebee, you're about this far away. And so that's why, they're being pretty mindful. If you want to get really good photos of them, you get a little bit closer,
Mm-hmm.
Those tiny bees without a macro lens, I mean, you even still have to get really close. And so obviously that's going to scare them away, this big phone coming at them or camera.
Yeah.
And then if you're wearing a hat, keep those little macro lenses. It just clips on real fast. I'm able to do it that quickly. But then I have to get really close to those little bees.
Right. Now the 10 has a shorter focal length. So the 5, I haven't tried the 5. I need to order some and try it out and see if it works.
Yeah. If you move slow, if you approach in real, if that little tiny bee is, busy collecting pollen or nectar and you move in real slow, you can get in there and get them. That's how I get some of those good shots of those little bees.
Yeah. And James is, probably got more practice on it than not probably. James has more practice on it than I have, and more experience with it. So he's really should be leading that workshop. So James, you want to lead that workshop this year? That'd be great. No, but in seriousness, I would like to have you there because I think you have great recommendations.
I feel like, oh, man, I really can't wait till we can get back to in-person workshops.
Yeah, I know. And we'll do more. Maybe we need phone cases that look like flowers. Yeah, that's a good idea. That's a really good idea, Cindy. Maybe we should send that out. We should send everybody we don't have the money for that, but it'd be cool if we did. You were Tina said she was referring to a postseason evaluation of the program. Anyway, I'm still not entirely sure I'm understanding that question. We did do a postseason evaluation that we sent out. It's actually still open. If you haven't had a chance to fill it out, you could still fill it out if you wanted to. But yeah, we do try to get feedback from folks and are happy to hear feedback. Species of bumblebees that we all observed, even better habitat. Oh, I guess that's something that we could also, one of the things that you can do that's kind of fun in iNaturalist is go to your location too and just see all the bees that you've actually taken photographs in your location, which is pretty fun. You get your bee list and all that. Yeah, did I miss anything? I don't think I've missed any questions so far. Any final questions or thoughts? No? All right, well, send me an email if you're interested in participating in any way, or just getting on the listserv if you just want to hear updates. Yeah, let me know. And if you don't, no worries. Have a wonderful holiday and a new year, and stay safe.